Good morning, everyone. I am pleased to welcome you all today. Today, we will be discussing the calibration of pH meters. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this presentation, we'll explore the importance of accurate pH measurement, the calibration process, and best practices to ensure precise results. I look forward to sharing valuable insights with you all. Let's get started. Today, I want to talk about our main objective, which is to ensure that pH measurement is accurate, reliable, and compliant with regulatory standards across all ranges, whether acidic, neutral, or alkaline. This is crucial because pH plays a significant role in many aspects of product quality. For example, it directly impacts product stability, which is essential for shelf life and effectiveness. It also influences drug solubility and how well a medication dissolves and becomes bioavailable in the body. Additionally, pH can affect the efficacy of preservatives, helping us prevent contamination and spoilage. Because of these factors, pH measurement is considered a critical quality attribute, or CQA, meaning it's a key parameter we must monitor carefully to ensure product consistency and safety. Throughout this presentation, we'll explore why precise pH measurement is so vital and how it ties into our overall goal of delivering high-quality, compliant products. Let's talk about why a 5-point calibration is necessary. Using just a single or even 2-point calibration simply isn't enough when you're working with samples that cover a wide pH range. A 5-point calibration helps verify the linearity of your pH meter, ensuring that the response is consistent across different pH levels. It also confirms the accuracy of your measurements in the acidic, neutral, and alkaline regions, so you're confident in your results no matter where your sample falls. Another crucial benefit is that it minimizes slope and offset errors, which can otherwise lead to inaccurate readings. Now, calibration in general is about adjusting your pH meter using standard buffer solutions. This process establishes a relationship between the electrode potential, measured in millivolts, and the pH value you're trying to determine. By doing this, you ensure your meter provides accurate measurements within the specific range you're working with, which is essential for reliable analysis. Let's take a moment to look at some of the key regulatory and guideline references that underpin our work with pH measurements. First, we have USP 791 pH, which provides specific standards for pH testing in pharmaceutical environments. Then, there's ICH Q2 R1, which focuses on validated analytical measurements, ensuring our methods are reliable and reproducible. We also follow WHO GMP guidelines, which are essential for manufacturing quality medicines globally. Similarly, the EU GMP Annex 15 offers guidance on validation and qualification processes within the European Union. Additionally, ISO IEC 17025 sets the standards for laboratory competence and quality management. A critical point to remember is that pH meters are considered critical analytical instruments. Their accuracy and reliability are essential because they directly impact the quality of our products. Ensuring compliance with these guidelines helps us maintain high standards and supports the overall integrity of our analytical processes. Let's take a closer look at the typical 5 calibration points we use in pH measurement. These are standard points that help us ensure our pH meters are accurate across a range of conditions. Firstly, we have pH 2, which is a strongly acidic solution. It's useful when you're working with highly acidic samples. Next is pH 4.01, which is acidic but less extreme, often used as a mid-range buffer. Moving to the neutral point, we have pH 7, which is essentially pure water under standard conditions. It's a critical reference point in many measurements. Then, we have pH 9.21, which is alkaline or basic, common in various biological and environmental samples. Lastly, pH 10.01, which is a strong alkaline solution, used for calibrating instruments that need to operate in high pH environments. It's important to note that all these buffers are traceable to national or international standards. This traceability ensures consistency and reliability in your measurements, no matter where you are or what you're analyzing. By regularly calibrating with these standard points, you make sure your pH measurements are accurate and comparable across different labs and applications. This consistency is fundamental to maintaining quality and confidence in your results. Let's go over some key calibration requirements to ensure accurate pH measurements. First, always use fresh buffer solutions. This helps maintain their stability and ensures reliable calibration. Next, temperature equilibration is crucial. Make sure your samples are at 25 degrees Celsius or that ATC, automatic temperature compensation, is enabled. Temperature variations can significantly affect pH readings, so controlling this is essential. Also, ensure your electrode is clean and properly hydrated before calibration. A clean, moist electrode provides consistent and accurate responses. Equally important is avoiding cross-contamination between buffers. Using fresh buffers for each calibration step prevents contamination that could skew your results. Finally, when calibrating, always go from the lowest to the highest pH buffer. This standard practice helps maintain electrode consistency and accuracy across the pH range. Keeping these points in mind supports the overall goal of precise and reliable pH measurement in your work. Let's go through the acceptance criteria for USB practice. First, when we talk about the difference between the buffer value and the observed value, a typical pharma acceptance is within plus or minus 0.05 pH units. This ensures our measurements are accurate and consistent. Next, the slope of our calibration curve should fall within an acceptable range of 95 to 105%. This range indicates that the electrode's response is reliable and linear across the pH span we're working with. Then, there's the offset at pH 7, which should be close to 0 millivolts. Generally, an offset within plus or minus 30 millivolts is acceptable, ensuring our electrode isn't drifting significantly from the true value. Now, for the calibration procedure, we start by switching on the pH meter and allowing it to stabilize, which is key for accurate readings. Next, we rinse the electrode with purified water and blot it dry to remove any contaminants. We then immerse the electrode in a pH 2 buffer to calibrate, followed by repeating the process with buffers at pH 4.01, 7, 9.21, and 10.01. It's important to record the observed pH values at each step carefully. Finally, based on these recorded values, we decide to accept or reject the calibration, making sure they meet the established criteria. These steps are vital to ensure our pH measurements are precise and reliable for our pharmaceutical processes. Let's take a closer look at the calibration data. As you can see, calibration is a crucial step in ensuring our measurements are accurate and reliable. Here, we focus on the key points that help us understand how well our sensor or system is performing. The data allows us to compare the actual values against the measured ones, highlighting any discrepancies. This process helps us identify and correct biases or errors. By analyzing this calibration data, we can fine-tune our system for better precision. This step is essential to maintain the overall integrity of our project, ensuring that our results are both valid and reproducible. Remember, good calibration underpins the entire accuracy of our work, connecting directly to the core theme of reliable data collection. Let's take a closer look at how to calculate the slope with this example. The slope, as you know, essentially measures how steep a line is. It's calculated by finding the change in the y values divided by the change in the x values between 2 points on the line. Here, we'll identify 2 points on our graph, note their coordinates, and then apply the slope formula. This process is fundamental because it helps us understand the rate of change between variables. Remember, accurately calculating the slope is key to analyzing the relationship between the data points and interpreting the overall trend. So, by working through this example, we reinforce how to apply the concept practically, which ties directly into our broader discussion of data analysis and trend identification. Now, let's talk about the linearity across the entire pH range. As you can see, our 5-point calibration confirms that the electrode response is truly linear. This means that the measurements we get are consistent and proportional across the full spectrum from acidic to alkaline conditions. This is especially critical when working with formulations that have variable pH profiles, where accuracy at different points can be a real challenge. Ensuring this linearity gives us confidence that our measurements are reliable, no matter where they fall on the pH scale, which ultimately supports precise formulation and quality control. Let's take a look at some of the common causes of calibration failure. First, old or contaminated buffers are a major issue. When buffers aren't fresh or have been contaminated, they can lead to inaccurate calibration results. It's essential to always use fresh buffers and store them properly. Next, a dry or damaged electrode can cause problems because the electrode needs to be properly hydrated and in good condition to provide reliable readings. If the electrode has cracks or is damaged, it won't function correctly. Temperature mismatch is another key factor. Since electrodes are sensitive to temperature, calibration must be performed at the same temperature as the sample to ensure accuracy. Inadequate rinsing between measurements can also introduce errors, so it's important to rinse thoroughly to remove any residual substances. Lastly, an aging electrode with a low slope indicates that the electrode is losing its responsiveness over time, which means it may need to be replaced to maintain calibration accuracy. These factors all tie into our overall goal of ensuring precise and reliable measurements, so paying attention to them will help maintain the integrity of your calibration process. Let's take a closer look at documentation and compliance when it comes to calibration records. It's essential that these records include several key details to ensure accuracy and traceability. First, we need to record the instrument ID, so we can always identify which instrument was calibrated. Then, buffer details and expiry dates are crucial because buffers can degrade over time, affecting the calibration. Temperature is another vital factor, as calibration conditions can influence results. Observed values must be documented carefully. That's the actual reading obtained during calibration. We also need to note the slope and offset, which are critical for understanding the instrument's performance. The acceptance decision is the final step, whether the instrument passes or requires adjustment, and this should be clearly recorded. Lastly, the analyst's signature acts as a verification that all these details are accurate and complete. Now, why is this so important? Well, maintaining thorough records is mandatory for audits and ensuring data integrity. These documents not only demonstrate compliance, but also support consistent, reliable results over time. In the context of our overall process, proper documentation underpins trust in our calibration procedures and ultimately helps us meet quality standards. Let's take a moment to review the key points from this slide. First, a 5-point calibration is crucial because it ensures accuracy, linearity, and compliance in our pH measurements. This means our readings are precise across the entire pH range we work with. Speaking of which, it covers the full pH working range, so no matter what sample we're testing, we're confident in our results. This thorough calibration supports reliable quality control decisions, which are vital for maintaining product standards. Additionally, proper full-range calibration is not just best practice. It's essential for meeting GMP, USB, and other regulatory requirements. So, in summary, accurate pH measurement starts with proper calibration across the entire range, and doing so helps us stay compliant, consistent, and confident in our testing processes. For further support, please don't hesitate to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention today. We appreciate your interest and look forward to assisting you further.